Thanks everyone for coming, and thanks to the CGSI organizers for inviting me. I think historically we started with like a little introduction slide, so I just wanted to say hi, I'm Erin. I did my PhD at Illinois with Tandy Borno, who's here, and also Bill Robb. And then I was out here at UCLA for my postdoc working with Sriram Sankara, and now I'm an assistant professor in computer science at the University of Maryland, where I'm lucky to work with these amazing students. Okay, so today what I'm going to be talking about is giving you an overview of a new technology called lineage tracing. Then I'm going to talk about how cell lineage trees are reconstructed from this type of data and why it's still challenging. And then I'm going to introduce constrained dynamic programming, which is a technique you can use in this context. And I'll tell you about some open problems. Okay, so first off, I think we got from Palash's talk, and many of you know, right, cell division results in a bifurcating process. And so, you know, as cells divide into daughter cells, we end up with this kind of population of cells in a tissue sample. And this is what we're collecting data from. And a major goal here is often to reconstruct the cell lineage tree. And we want to do this because cell lineage trees are useful. If you want to study how cells differentiate into different types, it can be really advantageous to have access to the tree. You could also use this to study tumor evolution. Okay, so cell lineage tree reconstruction is a first step in many types of analyses. And in the last talk by Palash, the focus was really on somatic mutations that were occurring in these cells. And that works great under some contexts, but it's not always going to work for your research questions. A big one here is a somatic mutation rate and time scale. And so if you're actually studying developmental processes and looking at how an embryo changes and differentiates into different cell types, you're not going to have enough somatic mutations to reconstruct the cell lineage tree. And then, of course, Palash also already mentioned, you know, that single cell sequencing technologies do have limits. The data can be very sparse, and it can be difficult to gather somatic mutations across the whole genome. Okay, so there's a number of challenges to gathering data to reconstruct cell lineage trees. And so in recent years, there's been this really amazing work to use CRISPR technology to induce mutations that can be used to reconstruct cell lineage trees. Okay, and so I'll just give you the high-level idea here. The idea is that you're going to introduce CRISPR barcodes into the DNA of cells. So here we have a barcode of 3 target sites. The introduction of the Cas9 enzyme is going to basically induce DNA cleavage at a target site. When the DNA repairs itself, it can introduce a mutation, and this mutation can get inherited by the daughter cells, and it's also going to prevent further cleavages at that site. And then, because you put the barcodes in, you can basically ensure that you can capture this data with single cell sequencing. And then you take your data and you're going to encode it as a character matrix, just like what Palash just showed you, where 0 is the unedited state. So for the red target site, we have a 0 because it was never edited. And for the purple target site, we have a 1 because the cell is carrying this edit. And you would just use positive integers, for example, to code up different edits. Okay. So now, you know, I've glossed over all the hard work in a wet lab and all the effort you need to do to get this technology to work and gather your data, which can take many, many months. But let's assume that this has all gone amazing. Now what you do is you have this lineage tracing data and you want to reconstruct cell lineage trees. And so this, you know, method development for this problem has really gained a lot of attention in recent years. It actually started back like at the turn of 2020. This paper came out in 2021. It's the Dream Challenge, and it was basically a challenge to get a lot of method developers to develop new methods for reconstructing cell lineage trees. And around that time and since then, there's been so many different methods developed and they work in different ways. Parsimony is what I'll be talking about today. There's many methods of this form. There's also likelihood and model-based methods, both Bayesian and maximum likelihood. There's combinatorial methods based on triplets and distance-based methods. And so any kind of method for phylogenetics, you can see being applied in this context in recent years. Okay, but today what we're going to be focusing on is parsimony. And so for those of you outside of phylogenetics, maximizing parsimony is minimizing edits, and is this core idea that the simplest solution is the best. And so many of the leading methods for cell lineage tree reconstruction are based on parsimony, and they do so by thinking about the properties of induced mutations. And so what I've drawn here is a cell lineage tree. This is the root cell, and there's two target sites at it. And so the first thing I want to note, right, is that the edits at target sites are heritable and irreversible. So on this edge of the cell lineage tree, the unedited state gets edited, and we're coding it up as a 1, and then it gets inherited by the daughter cells B and C. Now, DNA break and repair, you're going to actually have some repair edits be more common than others. And so this means that you can actually have a scenario where the same edit occurs at two different parts of the tree in parallel. This is homoplasy. And so we see that cell A and cell D are both in edit state 2, even though they're not descended from, even though they're not in a subtree together. Palash mentioned, right, that single cell sequencing can have dropout, which introduces random missing data. So I've hidden some of the values of the cells because we can't access them. And then lastly, you can have silencing at the target site, where essentially CRISPR's like cutting out a target site. And then this is going to give you heritable missing data because once the target site is silenced, it's going to be missing in all of the descendant cells. Okay, so the idea is that we have these kinds of sets of rules about what types of mutations are allowed, and then we want to basically reconstruct the cell lineage tree with parsimony. Okay, and so for those of you who are familiar with these types of approaches, you've probably heard of the small and large parsimony problems. In this context, the small parsimony problem, and I'm calling it the star homoplasy parsimony problem. This is a term coined by Palash. Henry Schmidt and Ben Raphael are also on this paper. It's basically used to denote that we're obeying the rules that I just told you about in terms of what edits are allowed. And so the small problem says that we want to assign states to the internal nodes of the cell lineage tree T, so that the cost of characters in our matrix evolving according to our rules is minimized. And so basically the cost is going to be the number of edits that are occurring, and they're going to be weighted by their probabilities, and we'll impute missing values while doing this. And so here you're just going to assign states to the non-leaf nodes to minimize the cost. Okay, so the small problem is it's polynomial time. Of course, we don't typically have a cell lineage tree, so we want to solve the large star homoplasy problem. Here, our goal is to seek the cell lineage tree whose leaves are labeled by the rows of the matrix or cells, and then this problem is NP hard. Okay, and so basically, since it's an NP hard problem, there's all of these techniques that you can leverage in this context. You might be familiar with branch and bound as an approach for parsimony. This is great because you have a nice optimality guarantee, but it's not going to scale to data sets with large numbers of cells. Integer linear programming is really widely used. A lot of The methods presented in the last talk are based on ILP. Again, great optimality guaranteed, but doesn't always scale to large numbers of cells. Very popular is heuristic search. The idea is you're going to edit a cell lineage tree, score it, and then, hill climb to find trees of higher scores. You know optimality guarantee here. And it is scalable because you can give up, take the tree that you have, but how long it runs is going to depend on your stopping criteria. And then what I want to talk to you about today is this technique called constrained dynamic programming. And here, the idea is that you're going to define some constraints that are going to restrict what the cell lineage tree can look like. And this is going to give you a limited but interpretable optimality guarantee. So you're not guaranteed to find a global solution, but you'll have at least some guarantee. And how scalable it is going to depend on how many constraints you have. And so basically this allows you to sort of balance the trade-off between finding an optimal solution and being fast. Okay. And so then the final thing I want to mention before I move on to this technique is that this type of data is really plagued by phylogenetic terraces. Is this idea that there's many equally parsimonious cell lineage trees. And this occurs for a number of reasons. One, there's technological limits to how many target sites you can currently add, or also cost limitations. And so typically you're going to have way more cells than you have sites, like these long, skinny matrices. Also, depending on the timing of how the Cas9 enzymes introduced, all the target sites could saturate like close to the root, and then a lot of your columns in the matrix aren't going to have many edits in them. They'll be invariant. And then if you have a lot of missing data, this is also going to make it hard to learn about the solution. And so this is one intuitive way to think about this. Two is that if you have a very large tree, not that this one is so large, I've labeled the branches that don't have mutations in red, and I would label the branches with mutations with numbers corresponding to the mutation in the matrix. And if we can track all the red branches, we get this tree with polytomies, or out degree greater than 2. And so basically you could resolve these polytomies in a variety of different ways, and they're all going to have the same parsimony score. And so you just get large numbers of optimal trees, both because of this contraction refinement situation, and also just because there can be many optimal solutions regardless. And so this is a big issue here to deal with that we'll come back to later. Okay, so basically now I'll just explain the intuition behind constrained dynamic programming for phylogenetics and show you some results. Okay, so the intuition here is that a cell lineage tree can be described by its induced clade. What is a clade? If you look at an internal vertex in the tree, you can see all the descendant leaves below it, and that gives a set. In this case, it's BC, and we call that the clade induced by this vertex. And so we can actually look at all the vertices of the tree and write down the clades induced. And I've highlighted in purple, blue, and red the non-trivial clades associated with their vertices. This clade of everything always goes to the root, so that's going to be on all trees with A, B, C, D, E in the leaves. And then these singletons are associated with the leaves, so they're also trivial. Okay. And so there's this relationship between a tree and its clades. We can use clades to constrain the search space. This is an idea from Hallett and LeGuerin at Recon 2000, and it's been used by Bryant and Steele, and also by Siavash Mirov and Tandy Borno in the Astral method. So it's been a very popular idea in phylogenetics. And to give you some intuition, on 6 leaves, there's 945 rooted binary trees. But if I write down this clade set sigma Only 3 rooted trees draw their clades from it. Put them here. If I have this character matrix and I care about the star homoplacy score, I can see that these, whoops, okay, there we go. I can see that these 2 trees have score 7 and this one has score 9 if all edits are weighted equally at weight 1. And so then, basically, I could take the 2 solutions here and build their strict consensus, which essentially only includes the clades of equally scoring trees. Okay, and so basically this just shows that there was a large number of trees if we allowed all topologies, but if we restrict the clade set, we reduced it substantially, and that could help a lot. And so basically what we do is we just add in this set sigma of clades, and then we require our output to the star homoplacy parsimony problem to draw all of its clades from that set. Now, the biggest question you probably have at this time is where on earth does sigma come from, right? Because we don't know it. And so, probably the easiest way to get it is to combine this approach with heuristic search. So basically, if you run a heuristic search, you could store all your best scoring trees or random trees, and then you could extract out their clades. You could also run different methods based on likelihood or distances or other techniques that save those trees. And then finally, you might have a priori knowledge. You might have done a visual analysis where you could identify some clades. You could have done clustering. Basically, if you know something about your problem and can encode it up as clades, you can enter it into this set. And then there's some existing techniques you can use to extend sigma to make sure it has properties where you can reconstruct cell lineage trees. And so basically, after you build sigma, now you have all of the inputs you need, and you just need to see the output. So how do we do this? We do this with dynamic programming. That's a method star CPP developed by my student Junyoung Dai. Dynamic programming, right? You want to find a solution by solving subproblems. And what makes this work? Well, the idea is that actually the clades constrain the state assignments at the internal nodes. So this red internal node highlighted is for the clade A, B, C, D. I've labeled it the state 1. And you can see that even when we change the subtree for this clade, it doesn't change the label. And you can prove that basically the clade is all you need to know what the internal labeling is. And that allows you to basically solve the large star homoplacy problem with dynamic programming. And this is the recurrence for people who are dying to see it, and the time complexity for people who are dying to see it. But the key is that this is polynomial time, and it takes a small amount of time on top of the heuristic search that you're doing. Okay, and you can also do pre-computation to kind of speed this up. Okay, so the last thing I'll note is that once you do this dynamic programming, you have a matrix, and you can actually use your dynamic programming matrix to find the consensus of all solutions that lie within that constrained search space. And to do that, you just need to count the fraction of solution cell lineage trees that induce each clade A. And you can do this without increasing the time complexity and having some increase in storage complexity. And then lastly, you could even use that dynamic programming matrix to sample from the place of solution, the space of solutions. And so we basically added this on top of the method. And I'll show you how this works in just a second, but the last thing I want to pose is a more philosophical question, which is, should we be reconstructing a binary tree at all? Because I've already told you, right, that there could be many mutationless branches in the tree. And the reason we do this is because if you allow polytomes in the tree, it makes the problem NP hard. And so even though philosophically we may not want to construct a binary tree, this is a necessary thing that we need to do to get a good running time. Okay. So lastly, this is nice in theory. We obtained a poly-time algorithm with an interpretable optimality guarantee, but is this any good in practice? And so to evaluate this, we did simulations, taking data from a prior paper where they start with a true cell lineage tree and simulate data under it. At the end, you reconstruct the tree, you contract the mutationless branches in both trees, and then you can compute tree error and accuracy and so on. And what we find are two things. First, as you would expect, the size of sigma that we're building depends on the simulation condition. And when you have 10 characters and 200 cells, the size is a lot larger than when you have 30 characters and 200 cells. And this is what you would expect, right? Because there's less data. And under that condition, this is where it looks like DP and taking the consensus has a lot of advantages. So we compare our method, star CVP, and taking the strict consensus of the solutions in the constrained space to ILP, and we see this lowers the number of false positive branches. It has a slight negative impact on the number of true positive branches, but then at the end, this means that we're higher in terms of the F1 score for accuracy. And importantly, we show that if we build the strict consensus from trees that we use to build the constrained search space, there were basically no false positive branches, but you missed a lot of true positive branches, and so that's why we're still better for accuracy. And then these methods are all basically taking kind of a single tree find via search or sampling from the solution space, and so there's a lot more false positives. Okay, and then finally we just checked the score, and we did see that sampling from our search space in this case is pretty similar to the score that you would get with the ILP-based method. Okay, we also looked at some biological data, and the brief takeaway I have for you there is that it's very hard to do a fair comparison. One of the things we did is we reconstructed cell lineage trees, contracted all the mutationless branches, and then refined with another technique, and then looked at the number of migrations and reseedings needed to, explain the data. We also looked at the score and the runtime. And we found that our method would run when startle ILP wouldn't. And in general, it got a reasonable number of migrations and a good number of reseedings, and was very good for SHP score. So we were getting reasonable results, but it can be a little mixed in terms of migrations and reseedings in terms of which method is best. And basically, even though we found fewer reseedings than other papers, I would sort of caution you in terms of these results because the refinement step probably plays such a big role. And so basically, to summarize, you can use constrained dynamic programming with heuristic search to get a limited but interpretable optimality guarantee and speed things up. And in simulation, this seems to be really great. And on biological data, it's difficult to draw strong conclusions because it is very difficult to kind of evaluate the quality of cell lineage trees in terms of migrations and reseeding. And so what we want to do here is try some newer methods like fast Machina that could help us evaluate the trees that we're getting in terms of migrations and reseedings. We want to validate more with the gene expression data. And then I think there's a lot of methodological questions that are open here, like how do you effectively build clade constraints? How can you prioritize solutions in the constrained space based on other objective functions? And will this work with likelihood? And then last, I just want to note, we get this to work with star homoplazy parsimony and Dola parsimony, whether you can do constrained dynamic programming with Fitch parsimony is an open question. So for someone looking for a math question, please solve that one. We want to know the answer to that question for our own interest. All right, and with that, I'll just say thank you to all of you and acknowledge Junior Dai, who led this study, as well as Ben Raphael and his lab, especially Jillian, Ewan, Palash, and Henry for helping us with the data and analyses. So thank you.